What we're going to look at now, it's the aerobic metabolism, which is the harnessing of energy stored in an ADH, FADH2, and using that energy to produce ATP. And that occurs through the electron transport in the electron transport chain, and it's coupling through oxidative phosphorylation. So our electron transport chain is located within the inner mitochondrial membrane of the mitochondria. And we have a series of membrane-bound electron carriers located in the inner mitochondrial membrane. So the mitochondria has two membrane, has an outer membrane and an inner membrane. And within the inner membrane, we have our mitochondrial matrix. The electron transport chain on the left, it's made up of a bunch of enzymes. And what the electron transport chain does, it takes energy stored within an ADH and FADH2 and uses that energy to transfer hydrogen ions across the mitochondrial membrane. And that generates a hydrogen ion concentration gradient that allows hydrogen ions to flow down gradient back into the mitochondrial matrix through ATP synthase. And as these hydrogen ions flow through here, they provide energy that allows ATP to be produced. So this is just a summary of what we just saw in the last slide. We have our outer membrane, inner membrane, and inside the inner membrane we have our matrix. We have the hydrogen channel, and then, as I mentioned before, with a high hydrogen concentration between the inner and outer mitochondrial membrane. Um, we have the inner mitochondrial membrane with the electron transport chain, and then that electron transport chain will pump hydrogen, protons, um, and inside the membrane we have a low hydrogen concentration in the matrix of the mitochondria. So let's go step by step through the electron transport chain. So the basic principle here is that the electron transport chain is made up of hydrogen ion pumps. And as electrons flow through these hydrogen ion pumps, they provide energy to those pumps that allows them to actively transport hydrogen ions and generate concentration gradient. The start of our electron transport chain is the complex 1 or NADH dehydrogenase. So NADH dehydrogenase accepts electrons from NADH to produce NAD. And it transfers those electrons. And as those electrons travel through complex 1, the energy associated with those electrons allows hydrogen ions to be pumped across the inner mitochondrial membrane into the intermembrane space. So NADH is becoming oxidized when it's losing electrons, and the electron transport chain is becoming reduced when gains electrons. So remember the rule from high school, Leon the lion says "Grr." So when NADH is losing electrons, we have the NADH becoming oxidized. And when electron transport chain is becoming reduced, it's because it's gaining electrons. NADH donates it is electrons at complex 1, whereas FADH2 donates its electrons at complex 2, which is called succinate dehydrogenase. So it functions the same way. FADH2 donate electrons and becomes oxidized, and electron transport chain becomes reduced. But complex 2 does not pump hydrogen ions. So importantly, when we start looking at how many ATP are produced per NADH or FADH2 molecule, we get less ATP from FADH2. And the reason for this is because FADH2 misses the first hydrogen pump in our electron transport chain. And as a result, we have the net number of hydrogen ions pumps is less for FADH2 compared to NADH. And we also, as a result, we have the larger the hydrogen ion gradient, the more ATP. So you get more energy or ATP produced per molecule of NADH. The next component of the electron transport chain is the ubiquinone, also known as coenzyme Q. It's an electron carrier. So this coenzyme Q or ubiquinone accepts electrons from complex 1 and complex 2 and transfer to complex 3. And the complex 3 is also known as cytochrome B, and it's another hydrogen ion pump. As electrons are transferred to complex 3, we get more hydrogen ions being pumped, and therefore increasing that concentration gradient across the mitochondria membrane. And eventually, those electrons are accepted by cytochrome C that carries those electrons to complex 4. And finally, the complex 4 is the final component of the electron transport chain. It's our third hydrogen ion pump. And again, as electrons are transferred through they give energy to cytochrome C oxidase. We get more hydrogen ions pumps, creating a great hydrogen ion concentration gradient. So importantly, complex 4 is a site of oxygen consumption within our mitochondria. So these electrons that are traveling through the electron transport chain have to be removed. So when there is an electron bound, the electron transport chain is reduced and can't accept another pair of electrons. So if we want continue electron transfer, continue pumping of hydrogen ions, continue ATP production, we have to remove electrons from the electron transport chain. And this is what oxygen does here. It accepts the electrons from complex 4. It becomes reduced to make water, and that allows electron transport chain to become oxidized and be ready to accept more electrons from NADH and FADH2. So if we had a situation where our cells didn't have oxygen, all of our electron transport chain would become reduced. They cannot accept more electrons and pump hydrogen ions. So it can produce ATP. So the production of ATP would stop. So that's the importance of oxygen. The electron transport chain can only carry one set of electrons at a time. And if oxygen is not present to accept electrons, the electron transport chain stops. So no oxygen, no aerobic ATP. The last component of ATP within mitochondria, it's a protein called ATP synthase. And remember that we have a huge hydrogen ion gradient across the inner mitochondrial membrane due to the pumps. So ATP synthase allows hydrogen ions to cross their inner membrane down their concentration gradient. So we have our hydrogen ions that have been pumped into the inner mitochondrial membrane from each of the 3 complexes. So we have a large hydrogen ion concentration gradient. And ATP synthase allows these hydrogen ions to flow down their concentration gradient back into the mitochondrial matrix. And as hydrogen ions flow through ATP synthase, they turn a crank that generates mechanical energy. And this energy is transferred into an ATP molecule. So this is similar to a windmill. as a wind blows past a windmill, it turns it, and this generates mechanical energy, which can produce electricity. ATP synthase uses this mechanical energy from the hydrogen ions, turning that crank to generate ATP. So the main idea behind this whole system is that we have electrons being donated from our reducing equivalents to the electron transport chain. So energy from electrons is being used to pump hydrogen ions out of the mitochondrial matrix into the inner membrane space, creating this hydrogen ion concentration gradient, also known as proton modal force. So when we have this gradient, now we can have hydrogen ions flow down their gradient back into the mitochondrial matrix. And as they flow down their gradient, they provide energy to phosphorylate ATP. So this animation, this video is just to represent what we just talked about. So we have a high proton concentration in one side of the membrane and a low ATP concentration. And this is the ATP synthase functioning. So the entire process is called oxidative phosphorylation, where we were using oxygen, we were oxidizing the electron transport chain, therefore phosphorylating ADP to produce ATP. So what the electron transport chain is, it's all about it's all about that. It allows hydrogen ions to move from mitochondrial matrix to the inner mitochondrial membrane, and that results in a large hydrogen ion gradient, so that hydrogen ions can flow back down their concentration gradient through ATP synthase into the mitochondrial matrix, where mechanical energy is generated as they move through ATP synthase, and then is used to generate ATP. So it's also important to remember that we need reducing equivalents in ADH and FADH2 because we need electrons being transferred to the electron transport chain. So we also need oxygen to remove those electrons so the electron transport chain can accept more electrons. So for each NADH molecule, we end up with 3 ATP, and for each FADH2 molecule, we end up with 2 ATPs. So less ATP is produced from FADH2 because it donates electrons to complex 2, because the complex 2 doesn't pump hydrogen ions. So it misses complex 1. So we have less hydrogen ions being pumped across the inner mitochondrial membrane, therefore a smaller hydrogen gradient. So this is a total ATP from glucose and glycogen. So we go from a small ATP directly from glycolysis and the TCA cycle up to 38 to 39 when the reducing equivalents are used by the electron transport chain. And the same thing happens for fatty acids. When reducing equivalents are produced in beta oxidation and TCA cycle are used in the electron transport chain, the ATPL goes from 9 to 148. So much more energy is produced when we use mitochondria to generate ATP aerobically through the electron transport chain. So that was the last step in our ATP producing pathways map. And that's it for this lecture.